Dear students, this is a topic of cell signaling. Today I am going to explain the overview over cell signaling. Cell signaling is key feature of life. Cells to sustain, to survive, to grow, to reproduce interacts with their external environment. Their external environment may be different in different cases. For an unicellular organism, a single cell has to perform all the functions related for the growth and its reproduction. It has to interact with the external environment, which is a non-living environment. In case of multicellular organism, a cell may interact with another cell. And in multicellular organism, the coordination between different cells of the body is essential for coordinate growth and development of that organism. So we can say no cell can live in isolation. In all multicellular organisms, survival depends on an elaborate intercellular communication network that coordinates the growth, differentiation and metabolism of the multitude of cells in diverse tissues and organs. Cells within small groups often communicate by direct cell-cell contact. We will examine how cells communicate by means of extracellular signaling molecules. As the slide is representing that in case of multicellular organism, the coordination between different organs is must. The coordination between tissue is essential. So how this coordination is achieved? This coordination is achieved by cell signaling. Now in cell signaling, obviously there is a signal molecule. The signal molecule is released by one type of cell, and the signal molecule act on other type of cell. The cell which release a signal molecule can be called as signal producing cell, and the cell on which that signal gets bind and display its signal is known as target cell. And in the entire signaling process, the cell signaling process, the signal molecule can be called as ligand molecule. So these substances are synthesized, means the signal molecules are synthesized and released by signaling cell, and they produce a specific response only in the target cell that have receptor for a signaling molecule. The response may be of diverse type, but broadly speaking, response may be the activation or inactivation of an enzymes which controls a particular metabolic reaction, activation or inactivation of certain type of proteins, activation or inactivation of transcription factors which further regulates the gene expression, activation or inactivation of other different type of protein molecules, change in membrane potential, change in the cell structure, cell shape achieved by cytoskeleton reassembling. So there are diverse different kinds of cellular responses. It depends upon the signal molecules. An enormous variety of chemicals, including small molecules like amino acid derivatives, acetylcholine, peptides, proteins, and even substance of lipid origin, they act as ligand molecules and are used in different type of cell-to-cell communication. The extracellular products synthesized by signaling cells can diffuse away or be transported in blood, thus providing a means of cells to communicate over longer distance than is possible by chain of direct cells to cell contact. So now another important question is that how the signal molecule will travel. For example, if the two cells between which the communication occurs, they are present distant or far apart. So how signal molecule will move? either that signal molecule will move to the neighboring cell, from that neighboring cell to another neighboring cell. This is not the way. So, in case of animals, this is achieved by the circulatory system. In case of plants, this is achieved by the phloem sap available in plants. Now, on the basis of distance traveled by the signaling molecule, that is ligand, we can categorize cell signaling into 4 different kinds or 4 different types. Here there are 3 types, it is mistakenly, but they are actually 4 kinds, and the name are autocrine signaling, paracrine signaling, juxtacrine signaling, and on endocrine signaling. It is based on the distance traveled by the ligand molecule, that is the signaling molecule. In autocrine signaling, the signal producing cell and the target cell are the same. The cell which produces the signal molecule, the ligand molecule, that ligand molecule binds to the signal producing cell itself and then generates a cellular an appropriate cellular response. So, in this case, in this diagram, you can see in red color, these are the signal molecules which are secreted outside the cell. These signal molecules then binds to receptors. Now, in important receptors play a very important role in cell signaling. Receptors and ligand must be compatible with each other, just like or we can say their shape, their structure are complementary with each other, just like the enzyme and its substrate. The interaction between the ligand molecule and its cell surface receptor or the cellular receptor is just like interaction of a substrate with its enzyme, and the interaction is actually a weak interaction. Weak interaction means either they will interact either by the signal molecule binds to the receptor either by ionic bond or by hydrogen bond or by hydrophobic interaction, but not by covalent bond. So, when it binds to the receptor, obviously there will be a change in the conformation of the receptor, and that change is transmitted to inside. We will study later. So, in this case, for example, many cells, just say for example cytokines and immune cells, growth factors. In this case, in case of immunology, or there are various examples of autocrine signaling. For example, a T helper cell, for example, it is activated by antigen presenting cell. Then T helper cell can release interleukins or cytokines. That interleukin will act on itself, and in response to that interleukin, the T helper cell proliferates by means of clonal selection. So, the clonal selection is the cellular response. Next is paracrine signaling. In paracrine signaling, the signal molecule acts on closely located cells, or we can say its neighboring cells. So, the signal producing cell first releases the signal molecule outside the cell, that is in extracellular matrix or extracellular fluid. From extracellular fluid, these signal molecule diffuses to the and binds to the target cell. So, this is what called as paracrine signaling. In this case, the signal producing cell and the target cells are different cells. There are various examples available for paracrine signaling. For example, synaptic signaling. In synaptic signaling occurs in nervous system between two nerve cells or between a nerve cell and a motor cell. So, in this case, in synaptical signaling, there is a synapse. So, presynaptic cell. The presynaptic cell releases neurotransmitter. Here, neurotransmitter is acting as ligand, and these neurotransmitters binds to post-synaptic cell, and they induces change in the post-synaptic cell, and the change is what? They generates action potential. Action potential means change in membrane potential. That is the cellular response. It's again a picture which is showing antigen presenting cell binding to a T cell receptor. The T cell receptor is here a cell surface receptor. So, when it activates that immature T cell, and immature T cells then releases cytokines or interleukins. The interleukins acts on other cells of the immune system. They can activate the T cytotoxic cells. They can activate another type of helper cells. So, this is what paracrine signaling, but they are neighboring cells. In juxtacrine signaling, the ligand molecule is not secreted by the signal producing cell, but it simply binds to the cell surface protein, or we can say here the cell surface protein of signal producing cell is acting as ligand. There are various transmembrane proteins which are present in plasma membrane of different kinds of cells, and they are also known as cell adhesion molecules, CAMs, like adenines, caderines, spot desmosomes, hemidesmosome. There are various different kinds of cell adhesion molecules. They bind to the neighboring cell, and this binding of these neighboring cells by CAM is a way of cellular communication. So, if a change occurs in a signal producing cell, that change is passed to the neighboring cell. For example, in smooth muscle cells or cardiac muscle cells, the collaborative contraction of cardiac muscle cells is responsible for pumping of blood inside the blood vessels. So this collaborative contraction and relaxation of cardiac muscle cells is achieved by such juxtacrine signaling. In endocrine signaling, which is common in animals, and the same type of signaling in plants is achieved by phloem sap. In endocrine signaling, the signal producing cell and the target cells are present far apart. So, ligand molecule has to cover a long distance to reach its target cell. So, the question arises, how ligand molecule reaches to the target cell? The ligand molecule reaches by means of circulatory system in case of animals, because it is the most fastest way of communication. So, a signal producing cell, popularly known as endocrine cell, release that ligand molecule, and in this case the ligand molecule will is actually acting as an hormone. So, hormone by means of blood vessels, it arrives or it reaches to the target cell, where it binds to the cell surface receptor and generates its signal, endocrine signaling. Now, once the signal molecule or the ligand approaches to the target cell, now what happened next? So, this is the main portion of cell signaling, and the very basics of the cell signaling was discovered by E. Southern experiment. E. Southern was studying the effect of epinephrine on glycogen. He was studying the effect of epinephrine on liver cells, that is glycogen metabolism. And in that experiment, he deduces very basics of the cell signaling. So, what happens in target cells? In target cells, cell signaling can be categorized as 3 different steps. We can study cell signaling in 3 different steps. So, and these steps are signal perception, signal transduction, and cellular response. So, once the ligand molecules reaches to the target cell, it first binds to the cell surface receptor. This cell surface receptor, and this step of binding of ligand molecule to the cell surface receptor, or it may be an intracellular receptor. So, binding of a ligand molecule to receptor molecule present on the cell or inside the cell, that process is known as signal perception. So, signal perception is simply the binding of ligand molecule to the cell surface receptor or the intracellular receptor. So, once on the target cell ligand molecule binds, the step is known as signal perception. Later on, this binding actually induces some changes in the cell surface receptor, and that change is then transmitted to inside. So, in this case, you can see in red color, the red color structure is receptor protein. This receptor protein then activates A protein, then A protein activates the B protein activates the C protein. All these proteins or the compounds are present in cytoplasm of the cell, of the target cell. So this step is known as signal transaction. So how this signal is transducing? So A protein activates the B protein activates the C, and the C protein then activates either metabolic enzymes, they can activate cytoskeleton proteins, they can activate transcription factor, and in this manner, an appropriate response is generated. One important feature of signal transaction is signal amplification. How? For example, insulin molecule or any other molecule binds to its cell surface protein, cell surface receptor, then it activates a molecule. It might be possible that this receptor protein can activate more than one A protein. For example, it activates two proteins. Two A proteins can further activate four B proteins. Four B proteins can further activate 8 C proteins. So in this manner, the signal is amplified, and these 8 C protein can activate more than 16 or 20 different kind of enzymes. So if more than 20 or different kinds of enzymes are activated by a single ligand molecule. So this is what called as signal amplification, and then it gives appropriate cellular response. So this main receptor protein molecules are either cell surface receptors like tyrosine kinase receptors or G protein linked receptors. They may be cytoplasmic receptor for ligand molecule which can freely cross the membrane. Generally, in this case, the ligand molecule is of lipid origin. Signal transition pathway. In signal transition pathway, protein kinases are the main components, or in some cases, second messengers like CMP, CGMP, calcium, protein kinase A are the major components. Thanks.